So here is our first real unit in our chemistry of life. And in section 1, we are going to talk about proteins. So proteins are organic polymers, which just means that they are carbon-based, and polymers means that they're made up of many parts. And those many parts are amino acids that are linked together in a specific order. And to function properly, each protein has to be folded into a specific 3D structure. And so the building blocks of proteins are amino acids, and they are organic molecules that have both an amino group, so now we know what those are, we know what amines are, and an acidic carboxyl group, which is carboxylic acid, and we know what that is as well. So the general structure is here on the screen. So each amino acid has a central carbon atom, which you can see right here. There's your central carbon atom. And it has 4 groups that are arranged around it. So you have your amine group over here, and then you have a carboxyl group over here. You have your variable side chain, which has R, which means that it's a most likely a hydrocarbon, but it could be a hydrogen as well. And then on the other side, we have our hydrogen atom. And the structure of an amino acid, the amine group and the carboxylic acid, provide convenient bonding sites for linking amino acids together into the protein structure. So since they have this amine group and a carboxylic acid, 2 amino acids can combine to form an amide. And in that process, they release water, which is called a condensation reaction, fittingly so. So you can see 2 amino acids side by side here and here, and this carboxylic acid group of the left amino acid is going to bond to this amine group on the right hand side amino acid, and in doing so they're going to bond here and here, which means that these 2 have to leave, and that's where you get your water for your condensation reaction. And then here you can see the formation of the amide that they're talking about, and that's how 2 amino acids become bonded, and that bond is called a peptide bond. So dipeptides are molecules that consist of 2 amino acids bound together by a peptide bond. And each end of the 2 amino acid unit in the dipeptide still has a free group, either amine group or a carboxyl group. And each of those can be linked to the opposite end of another amino acid, and the cycle continues and continues, forming more and more peptide bonds. And now a chain of 2 or more amino acids linked together by peptide bonds is called a peptide itself. Now, living cells always build peptides by adding amino acids to the carboxyl end of a growing chain. So they just continually go through these condensation reactions where the carboxyl group of one amino acid bonds with the amine group of the other, releasing water and creating an amide. And there you get your dipeptide, as you see in your picture over here. Now, a chain of 10 or more amino acids joined by peptide bonds is called a polypeptide, so many peptides. And then when a chain reaches a length of about 50, then it's finally called a protein. Now, there are only 20 different amino acids that form proteins, and that kind of makes you think that there aren't very many proteins because there are only 20 things that build them. When in fact there are about 80 to 100,000 different proteins that we know of. So the variable arrangements that they can make means that there are many, many different proteins out there. Now, as proteins are being built, those long chains of amino acids will start to fold into their unique 3D shapes even before they're fully synthesized or fully made. Now, they may create helices, which you can think of as coils, like my hair. They could form sheets, which kind of look like an accordion. And then, of course, while those are being built, it could also change direction completely and fold over on itself. Now, the shapes that they make, where and when, depends on the interaction between amino acids. And by interactions, I mean intermolecular forces, which we have talked about. Depends on what intermolecular forces are there. But the overall 3D shapes of many proteins is what's called globular, but that just means that they're kind of an irregular sphere. So once they get all those shapes together, they end up in this weird, awkward sphere. But some can be long, fibrous structures. And then things like changes in temperature, ionic strength, pH, and many other factors can disrupt those interactions between amino acids, the intermolecular forces, which can result in unfolding and uncoiling of the protein. And that's when denaturation happens. And that's the process where your 3D shape is lost or disrupted. And proteins only function when they are properly built, so in the correct order, and properly folded. So a denatured protein is an inactive protein most of the time. So that is why your body has a very strict pH range that it can survive in, because once your proteins start to denature, then a lot of processes no longer work. That's why we have to maintain a specific temperature so that our proteins do not become unfolded. So all of these things kind of come back around to biochemical factors. So just to kind of give you an idea of what protein structure is like. So you start off with your sequence of amino acids, then they start forming their shapes. So I've given you an example of the helix shape, the coils, and then the sheet shape. It's called a pleated sheet. You can see the accordion. And then in different parts of the protein, they'll have these different shapes, and so they'll fold up on a much larger scale to get a full 3D structure, like the third picture here. And then they will perform their function. And those functions are many. They can catalyze reactions, transport substances, regulate cellular processes, form structures, digest foods, recycle wastes, and occasionally serve as an energy source when there's none other available. So enzymes are a specific type of protein that have a specific function. They are biological catalysts, and a catalyst is something that speeds up a reaction without being used up in that reaction. And they have a specific way that they work. A substrate is the reactant or reactants in an enzyme catalyzed reaction, and they will bind to a specific site on the enzyme known as the active site. And once that substrate binds to that active site, the active site will kind of conform to that shape to make it fit tighter, which is called an induced fit. And the substance created by the substrate and enzyme is called an enzyme-substrate complex. And then that is then converted to product because the catalyst itself lowers the activation energy to allow a chemical reaction to occur and change the reactants into products. And then it will release your product, and then it will perform the same function again because the catalyst is not used up in the process. Enzymes and their shapes are very specific, so they're built for a specific function, so only those specific substrates will fit in the active site of that particular enzyme. So they're very, very specific, like a lock into a key. So other proteins are meant for transport, and they will transport smaller molecules throughout the body. The most well-known one, and the one that you will talk about in any college chemistry classes, is hemoglobin. That is a protein that is used to carry oxygen through your blood, and it uses a heme group, which you can see in my picture, and that's where the oxygen is carried. And you can see kind of its 3D structure here in your picture. Another type of protein is structural, and they will form vital structures like skin, ligaments, tendons, bones, hair, and fingernails. And those would be things you heard of like collagen or keratin. And then lastly, some proteins function as hormones, which are just messenger molecules that carry signals from one part of the body to another. Now, some hormones are proteins, some are not. But one example of a hormone that is a protein is insulin, and it's a pretty vital protein to your body. It tells you when the sugar in your blood is too high. So, we've also determined a way to artificially create proteins, and that's how insulin is made for diabetics. And there are other proteins that are synthetically made as well for medications and things like that. So we've kind of perfected a way to use proteins to our advantage at this point. So that's pretty much going to sum up section 1, all about proteins. So be ready for those review questions.